Good afternoon, everybody, and thank you to be here this time. I'm Noof Bani Khaled from Universiti Putra, Malaysia. Today I will present a part of my genomic study for detection of rare ST547 among multidrug-resistant Acinetobacter baumannii in Malaysian hospital. So just through my contents in this presentation, I will go fastly about my research methods, results, and discuss those results in briefly. So, as you know, Acinetobacter baumannii is a gram-negative coccobacilli. It is multi-drug resistance, recognized as major nosocomial infection, and it is the major cause of hospital-acquired infections. So, according to World Health Organization in 2024, Acinetobacter baumannii, especially carbapenem resistance, as a first and most critical priority in hospital settings. If you look at the right, there is a map just show how carbapenem resistant prevalent overall the world in Asia, Latin America, parts of Africa, and Middle East. If we go through just Malaysia, we can see there is increase, significant increase, especially for carbapenem, IMP, and MAM. So those carbapenem, around 60 to 70% increase since or from 2000 until 2016. This study, like a systematic review for many hospitals and medical centers in Malaysia. So the objectives of our study is to investigate the genomic antimicrobial resistance determinants, to find the plasmid profiles, phylogenetic relationship of Acinetobacter baumannii, those collected in 2021 in healthcare facility in Malaysia. I will go just quickly through my methods. Our methods to collect, first, we collect 55 samples. We took those medical records from the microbiology lab, and those data going just to take the Acinetobacter in general. So we didn't know what is the species specific for those samples. Then we did bacterial culture. So for those samples, we're doing two methods, genotypically and phenotypic. Then we compare the results. For the phenotypic, we use disk diffusion and MIC. We will not talk about it. And the genomic method. Through the genomic method, all samples go DNA extraction, PCR, 16 sRNA by Sanger sequencing, and go through the WGS. But not all those 55 go through WGS, just 37 samples. Those go through WGS. Why we choose it? We try to find the best DNA concentration and the phenotypic characteristics that we needed to further genomic. Those 37 go to nanopore platform. Then after we finish all nanopore, find 10 samples, low coverage. We try to repeat, but then we suggest to go through Illumina for short read sequencing. And after this sequencing, we go bioinformatic analysis to show to find the antimicrobial resistant genes. So for our results, the clinical and demographic data for those patients, we can see the highest percentage for male, and the samples of the sources from tracheoaspirate, which means the respiratory system. And 85% samples from ICU, those who's in our isolates. For AST profile, we can see here most of the sample resistance to resistant to antibiotics, except I think one sample who is sensitive to amikacin. For the genotypic results, as I said, we identified all the samples, 55 Acinetobacter baumannii. And we just choose the 37 who's made our criteria to further WGS. And we can see here the coverage up to 342x, and the genome size around 4 million. Okay. Now what we found, for the ST, all samples by Pasteur, ST2. By Oxford scheme, just one sample, 547, and around 50% for the rest of samples from 1816 and the other from 1806. Okay. Now the antimicrobial resistance profile, same with, mostly with the AST that we did. So you can see here, most of the samples, 100% resistance to carbapenem, resistance to gentamicin, to amikacin, and other antibiotics and aminoglycosides. This is just to show what is the resistant genes we found that distribute through the samples. So for aminoglycosides, most of them are resistant, as I said, but for beta-galactam, not all, just from each variant, one sample. This for all 37. For plasmid profile, it is interesting to see that 12 samples from those, 12 isolates, from those who go through nanopore, 12 are the same plasmid, and another 7 with other plasmid, I cannot see the name, and the other one, 117, also 5 samples. Those not found in literature. So we think that those plasmids, can we do further studies and further research in the future for those samples to see the resistance genes. The phylogenetic analysis, we can see here 5 clades for the Acinetobacter. The one we talk about, 547, just one sample, and others like clusters from 1806, 195, and also 1816 and 195. Okay. So to discuss further, this infection predominant among ICU patients and male patients, which is similar with other studies in Malaysia for other states, one in Kuala Lumpur and one in Terengganu. And most of the specimens from respiratory, which is also like other studies in China and near countries of Malaysia. Okay, and most of the resistant genes from aminoglycoside and beta-lactam. So WGS is still WGS driven by black oxa 23 consistent with its global dominance as other studies. And also the Oderia, there is absence of RND efflux pump genes suggestion. There is alternative resistance mechanism. So all the samples under ST2 which is the same result for other studies in Malaysia. For the plasmid, I said before, there is no lectures that go through those plasmids, so we will do further study later to investigate it. For the ST547, you can see here, there are just 9 samples over the world. 7 that count, 5 in China and 2 in Malaysia. So we think that suggests there is maybe an epidemiological link between those samples. Maybe because some of Malaysian people from original Chinese, or maybe some traveling through countries. The phylogenetic tree, we can see there is similarities between those isolates. So we suggest maybe there is an outbreak during that period for the collecting isolates. So in conclusion, this study just provides an insight to the resistance and also the clonal of dynamics of multidrug-resistant Acinetobacter baumannii. Still, WGS surveillance, can we investigate the resistant genes and find it? Also, for our detection of plasmids, we think also there is a horizontal gene transfer between those samples. Okay, so finally, just to acknowledge my committee in the university and the MLT in microbiology staff in hospital to assist us for this work. And hopefully we can do something new for the AMR. Thank you for listening.